Sanchez. I'm a student at United Tribes Technical College. I'm majoring in environmental science and research, and I'm working under my mentor, Dr. Hona, and we are conducting research or study on how to control or manipulate the iron concentration in wheatgrass. So we separate our samples into trays. We add the wheatgrass seeds, and these seeds we got them from Amazon, and throughout the 7 days watered them with water iron concentration. The height, the mass, and the number of plants, we cut them and add them into this ceramic, and we place them into the furnace. As a result, we get a white ash. We add about 5 drops of hydrochloric acid, and that dissolves the ash. Take samples of our solution. We add 5 milliliters of sodium acetate, 5 milliliters 110 phenanthrene, 1 milliliter of hydrosilamine, So I'm first adding the blank solution into the UVV spectrophotometer. We add that in there, close this, and we can run the program. Now that it has ran through, we take the sample and we add our iron sample. We run it again to measure the absorbency. It has a peak absorbency at around 511 nanometers. To test our sample solutions, we first have to add our blank solution. Every time you run a sample, you want to add your blank solution. As you can see, we are running it at 511 nanometers. There is 0 absorbency in the blank solution. We take this blank solution, replace it with our sample, and now we can run the solution. This sample has an absorbency of 0.799.
We repeat this process for each standard solution. This will give us a straight lined graph that will show us the absorbency at each concentration.
So after dissolving our ash in acid and adding our solutions, we're given a sample solution for the iron found in wheatgrass. Run it as a baseline. We replaced our blank solution with our sample. Again, this is supposed to measure the iron in the wheatgrass that we grew. Now we can see that at 511 nanometers, which is the wavelength, there is an absorbency of 0.264. Now we take this data and compare it to our standard solutions to measure the concentration in the wheatgrass. There have been many studies showing that wheatgrass serves as an anti-cancer remedy. Boosting the iron concentration results in the ability of moving more oxygen through blood cells. Wheatgrass is typically consumed in the form of juice. It has many benefits like boosting metabolism, promotes weight loss, has anti-aging benefits, and many more. I think that this study or research can expand to look into what other plants or resources have to offer. We can look at the elements and nutrients and try to enhance those so that these are available and are more beneficial to a consumer.
Hi, I'm Dr. Ram Hona from United Tribes Technical College, Bismarck, North Dakota. Sanchez, a student of mine, is working in iron concentration control in wheatgrass in my lab. We are interested in wheatgrass because it has different health benefits such as anti-inflammatory, anti-aging, anti-cancer, and others. Wheatgrass can be grown easily everywhere, such as in garden, home, field, and it does not require any specific environment or location. It is commonly taken as juice for supplementary purposes. We are fascinated by the anti-cancer property of the wheatgrass. It has been proved by research and clinical trial that a regular intake of wheatgrass juice can inhibit cancer cell growth and treat the cancer disease, particularly the colon cancer. Here we can see the red blood cells flowing to the veins along with the blood. Blood is composed of the red blood cells, white blood cells, and platelets. Now, red blood cells contains iron. Cancer cells grow when there is irregular cell activities, which is caused by the lack of sufficient oxygen supply to the cells. Oxygen supply in the body takes place through blood. Blood contains red blood cells, which contain iron. Iron is the main component in the blood to carry the oxygen to the cells throughout the body. Iron in hemoglobin combines with oxygen to become oxyhemoglobin and leaves oxygen to the cells, and the oxyhemoglobin then converts to hemoglobin. These are the chemical structures of hemoglobin, heme group. Now you can see iron at the center, which is not connected to oxygen. So this is heme, and this iron combines with oxygen to form oxyhemoglobin or oxyheme group. Now, when we surveyed in different tribal communities in North Dakota about the knowledge of wheatgrass juice, people from the different communities, nobody said that they know about or they heard about the wheatgrass juice and its health benefit. So it is the most important part here for tribal communities that they should know about the health benefits of natural products like wheatgrass or any other plants. And this is a natural way to take natural ingredients like iron and other minerals rather than to take direct chemical supplements. It is easy to grow and can be grown everywhere, anywhere, as we discussed before. It does not require any specific location or environment. It can be a subject of outreach programs to educate the community about the health concerns because most of the community members are not concerned about health and they have lack of the health education. And iron deficiency in the body of the communities can be fulfilled by natural ways of food taking such as wheatgrass juice. And for this project, I acknowledge my student, Sanchez, and to North Dakota F-Score, who granted me fund for equipment and then to the NSF. Thank you.